Okay, so for the next hour, I will talk about variant calling. I'll give you an overview for half an hour and then specifically talk about germline calling. And then we'll go into a hands-on exercise with those notebooks that you cloned. Aaron will walk you through variant calling with the HaplotypeCaller. You'll get to do it yourselves. We'll take a small break and then he'll talk about joint calling, which is bringing all your data together from all your samples. But if anyone has not yet, if the cloning didn't work or you have a problem, please let Aaron, Anton, or Emma know in the back so they can help you out so that when we do get to the hands-on exercises, everyone is ready to go. Okay? Any questions to start with? All right, in the other room, if you have questions, just come running over anytime. So this is now an introductory overview for variant calling in general, not just germline. And so there's a lot of basic concepts. I will start kind of at the high level and move into details for this presentation. The GATK can work on any organism, almost any organism. The human genome is diploid, which means you have 2 copies of your chromosomes, but plant DNA often has 4 copies. The GATK in theory can work on any organism, but it was developed for medical genetics, and in this workshop we focus on human genetics. So let's start with it. So I mentioned that human genome has 3 billion bases, and really you have 2 copies. So actually you have 6 trillion bases. Any 2 people in this room will have a lot of DNA in common. Anywhere between 99.5 or 99.9% in common. But that also means you have a lot of differences. So if you have 0.1% of your genome is different, but you have 3 billion bases, that still means you have 3 million differences. 3 million is a lot of differences. A lot of differences. Okay. Most of those differences are common. That means if I have that difference, if I have that mutation, that variant, then good chance most other people or many other people will also. Okay. But I'll also have some mutations that are uncommon, that are rare. Maybe I'm the only one in the world that has it. Okay. But most of your mutations will be common, like 99% or even more will be common. Yeah. Oh, I'm not sharing the screen. Oh, okay. Harry? How do I share my screen again? Why don't I have Emma in the room? You left me Emma. Thank you. And so what that last line is that most of those sites are biallelic. That means there's the reference version, the wild type copy, okay, and then there's one base difference. So again, if the reference has an A, there is one difference in the population. Maybe there's a T, but it's not like some people have a C and some people have a T and some people have a G. Those sites happen too. There are positions in the genome that are multi-allelic, but most variants that we know of are biallelic. The details on the slide, you can read them through if you want, but the most important thing is that we always look at sequencing relative to a reference. Human reference, a cow reference, a pig reference. It is not a person, but it's amalgamation. It's a combination of many people. The human reference, the original one, was actually 5 different people from different ethnicities. Africans, Asians, and Caucasians, I think, all went into making the reference. So there's no one person that actually has the exact same basis as that reference. But they did the best they could. They took the most common alleles, most common bases from each person, just to get something that's close enough to the canonical human genome. Okay, but the actual bases aren't important. It's really what differences you have relative to those to the reference. Okay, and as we go on time, they improve the reference. They add centromeres; those are the pieces in the middle of your chromosomes that are very repetitive, and telomeres on the ends. And there's very repetitive regions all over, and so they're improving them. There's actually a new reference that just came out like months ago, or weeks ago, called the T2T, the telomere to telomere, the end-to-end. where they think using long-read sequencing have gotten a complete categorization of the human reference. And for that, I can use certain samples that we'll talk about later. Okay, so you can look at that now, and actually, so I have a workspace I was mentioning where I've added that into my own reference, and so you can play with that too if you want. But for most of the work we'll do today, I think it'll be against what we call HG38. That's the most commonly used reference today. Aaron, yours is against HG38 today. Do you know?
It's against the number 4 that actually HG.
Actually, no. Okay, so Aaron's going to use no reference. So, oh well, thanks for you. Okay, so this is an important slide. So there are many different types of differences in your genome, lots of mutations. The most common ones are called SNPs or SNPs. SNP is single nucleotide polymorphism. So one base difference. And there are about 3 million of those in your genome. The next kind of category is an indel. It's an insertion or deletion. And usually it's a few bases that are extra or missing from your genome. And they're usually small, maybe 1 base or 2 bases or 50 bases, but not more than that. I think when Emma talks about structural variation, she'll define it more there, but generally it's around that number, between 1 and 50 bases. Then you can get much larger structural changes in your genome. So what we call CNVs, copy number variations, that's where you have a whole swath, a whole region of your genome missing, much larger than 50 bases, or you have multiple copies of it. And I'll show you some examples of this soon. And then the SVs are structural variation. So CNV is a subset of structural variation. Structural variation also includes lots of crazy things. Inversions, and I'll show you some examples of inversions where whole pieces of your genome are turned around or translocated, where you have 2 chromosomes that now connect. Okay, and so there's lots of different variant types, and you also have to look at it in the germline and somatic case. Germline means you inherited it from your parents. So one of your parents has, or both, have passed on the mutation gene. There are, in any new baby, there are about 18 to 20 de novo mutations. That means mutations that were not given to your by your parents, but most of the 3 to 4 million mutations you have are from your parents, and that's germline. Somatic is the cancer case. Just in part of this, of the process of biology, as your cells duplicate, there are changes that happen, and it's part of the evolutionary process. It's why science happens, but it also means that as your cells divide and replicate, they also introduce mutations, and those are somatic, meaning they're not inherited and they're not present in every cell of your body. They're only present in some, maybe one cell, or if that cell keeps that copy and divides and replicates, a few cells. And when those cells then misbehave, and we'll talk about that in the cancer section, that turns into a tumor. Okay, so for today we'll mostly we're focusing on germline. Somatic is going to be on Wednesday, I think. I'll try to give you a background a little bit here of both, but after this, I'll focus on the germline. Okay, so here's an example. If you're looking at IgV, IgV is something we've talked about, and we will continue to show you. Right in the middle of, on the left of the screen. Actually, the benefit of the people in the other room, I'll stick here to the browser. So on the left side, you see those eyes, those purple eyes. Again, that represents an insertion. So that's what it would look like in IgV. And maybe a little bit over to the right, you see the brown and blue column. Okay, that means there's both a G and a C, and the reference is not even that. Neither of them are in the reference because they're both highlighted. Okay, so that's what it would look like though when you have a heterozygous SNP. That means heterozygous is you have your genotype is different. It's not, there's one C and one P or one G. Right? They're not 2 C's. Versus a homozygous means both copies from your parents are the same allele, same letter. And that would be a SNP. Okay, a single nucleotide polymorphism where you have your bases changed, but there's no extra or missing bases. Now look at the stuff on the right, all that color. When Emma was talking about soft clip bases that don't align, this is usually what happens. is that part of the reads there are aligning, and then the ends of them are not. And if I mentioned those Q2s where the ends of the reads are not high quality, if that, if you see Q2, that the Q2 process happens randomly. But in this picture here, you see they're all happening at the very similar or same position. So that's no longer random. And there's usually some signal there. And that signal is used to find much larger things hidden in the DNA. And so when Emma talks about structural variation, she'll show you how they use that signal to detect much larger events. Remember, these reads are very small. The structural mutations are very big. It can be thousands or tens of thousands of bases long. You're trying to discover them from these little pieces of DNA. And so this is the kind of signal we use to find those bigger things. Someone had asked about how we represent variance. So here's the answer. We will go into this more detail later. I think Aaron's going to cover some of this also. So I'm just going to keep it high level. There's always a header section so the VCF is the variant call format. I'm actually an author for this format. Don't hold it against me. It was those days we had very few samples, so it doesn't really scale anymore, and we're trying to update it. But in the early days, we had a few thousand samples in the world, and now we have millions. And so hard to represent millions in this format. But we'll do it for now anyways today. But the important piece is the bottom section, where you start to list all the mutations. So you have just going through the fourth to last line tells you is like the header, the last header line, and then you actually have data. So this example, you have chromosome 20. The first record is position 14,370 on chromosome 20. Okay, we'll skip that E for now. The reference is a G. That means that's what the G has at that position. The reference has a G, and the alt is an alternate. That's the mutation. Someone has an A. Then let's skip to the end. Those last three columns for the IDs for those samples are NA00001, 2, and 3. We made up those numbers. Again, Aaron will go into details, but the section below shows you what their genotypes are. So a 0/0 is going to be homozygous reference. 0/1 or 1/0 is going to be heterozygous, and a 1/1 is a homozygous variant, as you pull copies of it. So in that first column, that first record, this row, NA00003 is 1/1, which means that he or she has 2 copies of the A. And we'll review this with you and kind of talk through some of this. I also say you can represent these large structural events in VCF format too. Now, again, they can be tens of thousands of bases long. So we don't want to put all 10,000 bases inside this file. That gets unwieldy. So we have these symbolic alleles saying, hey, it's a deletion, and here's the start and finish, but we're not going to put all the bases inside. All right, so let's talk through. So again, we have workflows, we have tools for almost all the classes. The structural variation on the somatic side is something we're working on, but nothing we have ready for you today. But in terms of all the other classes, Germline, and all the big and small mutations, we're going to cover that in this workshop. Okay, so I'm going to do some overviews now. I'll repeat some of this for the HaplotypeCaller, but I'm going to just give you a high-level overview of what you're going to see in the next few days. For, again, Germline, inherited SNPs, 1-letter changes, and indels, small insertions or deletions. The most important thing for this, and Aaron will cover this today at the end of the day, after your next break, he's going to cover something called joint calling. This is very important. Unless you're even in a clinical case, I can think, maybe you're looking for, you have one patient a baby who has some genetic disease, you're trying to figure it out. And you find this baby you've done sequencing, you find this baby has 3 million mutations. Right, which one is the one that's causing the disease? Which one tells you what's affecting the baby? You have no idea. So the only way to do that is to take all 3 million mutations and study them against what the rest of the world has. If you find a mutation in that baby that no one else has, and it's one of the genes, there's a good chance that's the one. But you have no idea if you don't look at that in the context of everyone else in the world or everyone else in the family. Okay, so looking at one genome at a time does not empower you to do really great science. And if you're doing especially larger, if you're going to do the Taiwanese biobank project, one day hopefully you'll do that. If you want to do that, you can't look at one person at a time. You look at everyone in the context of what the entire country has, or the entire world has. And to know that, look, this type of mutation is common, well, that's unlikely to be a disease causing if it's very common. Or to find the structure that is common within your population is very important. So jointly analyzing all your samples together is very, very important. And Aaron will talk about all those details later. So I'm going to skip this a little bit. This is, again, to show, Aaron will cover this too, but we have a special format for dealing with data that's going to be jointly analyzed. It's kind of DCF-like. So when I show you the HaplotypeCaller soon, there's a mode where we don't kind of fully make variant calls. The HaplotypeCaller goes from a BAM file, your reads, to variants. But in this mode, it's going to almost make variants. It's going to make something close to them, and that's what this is. And it'll still do all the math and all the smarts. Also, what's important is remembering that even though I mentioned that the sequencing data is not perfect, it's definitely not perfect. And we do our best to represent everything, to fix things and reprocess it into recalibration and remove duplicates and just get all the math right. And still, it's not perfect because the human genome is complicated. The sequencing data is imperfect. The mutations get complicated. So there are things that still it's hard to catch. But when you look at them with one sample, almost impossible. When you see the same systematic problem across many samples, now you're empowered to do something about it. So there's always filtering of the variance that we do downstream. After we do this joint calling and see all the data together, we can say, look, this thing looks low quality in this sample, but high quality in that sample. So maybe it was just, it's fine. It just wasn't sequenced well in this. But this variant is low quality in all 100 of these samples. That's probably an error. It's probably a mistake in the sequencing, not a real mutation. Okay, and so we'll show you examples. Tomorrow we'll talk all about filtering, and there's a lot of machine learning that we do now for this. It's very, very good project for machine learning. Also, so I'll say at the Broad in our production processing, we don't use this step because we have no idea which project's sample comes from. We don't know if the sequencing is from a family or whether they're Caucasian, European, Asian, African. So we can't actually run this. But anyone who knows where their samples come from, they can do something called genotype calibration, right? Refinement. And it is basically fixing yet the last set of errors we have in terms of deciding whether your genotypes are accurate, whether heterozygous or homozygous. And this is fun math that we'll talk about tomorrow. I'll show you some examples of that. And then lastly, it's very important to annotate all of your mutations with how much of an effect they have biologically or could have biologically on your patient. So just at a high level, sometimes you have an insertion or a deletion. Let's say a deletion of one base. If it's randomly in the genome, it probably doesn't do anything. But if that one base is a deletion of one base in an exon, remember the way it works with exons, you have these codons. Every three letters, every three bases in an exon translates right into gets translated into a protein. And if you're missing one of those bases, everything gets shifted. It's called a frame shift. And so that one base deletion can have a major effect on the entire translation of that gene, of that exon. Right? So we try to annotate every mutation with its potential effect on the body, on the genome. And that's very important, because if you're looking at the mutation that is potentially causing a disease, well, the ones that might cause a frame shift are more likely, as opposed to ones that lie in the middle of an intron. Again, we'll go over a lot of this tomorrow. I'm just giving you a high level. Okay, so we have a very, very standard pipeline. You'll do a lot of the steps with us in the hands-on for the next few days. And all of it is documented well. You can go online, you can look at the slides, you can look at the website. We have a lot of this. Somatic cancer is different. Now, there's always an argument between the folks from my team who do germline work and somatic work. Which one is harder? All right, so the germline folks say, come on, look, the somatic mutations are generally huge. It's so easy. All right, but the somatic folks will say, okay, well, I'm gonna, it's way more difficult to do cancer color. Okay, so why? So in germline, again, you're expecting half of your reads to show a mutation if you get one copy from your mom and not from your father. So it's a 50-50 ratio. Or if you get two copies of the mutation, 100% ratio, right? So it should be in every read. Cancer is very different. Remember, you're looking, when you try to extract a tumor tissue, well, you're not getting it, it's not just perfect. You get some of the normal tissue also, the non-tumor tissue. Also, every cell in the tumor is different because they're all expanding and growing and changing very rapidly. And so you have two types of contamination. You have your normal tissue is contaminating your tumor, and the tumor is so different. And so there's the signal to noise ratio is so massive in the cancer use case. So that's why the cancer team says we have a way harder problem than anyone else built on. We'll talk about this on Wednesday, but it's important. This is one of the most important concepts of the somatic, the cancer work to understand. One of the most important concepts. There's so much noise, and we try to do as much as we can to remove that noise. So the two types of noise when you're looking at it from sequencing is you want to remove the germline mutations. In the somatic case, I want to see which mutations are only present in the tumor. All the stuff you inherited from your parents are germline. I don't care about those. Those are in all my cells. So we try to encourage you to have a matched normal. So if you extract tissue from a tumor, also take a blood draw. And so you can compare the two and say, look, I can see that the mutations that are present in the blood, clearly they're not somatic. They're not part of the cancer. They're in all your cells. But that doesn't help you remove these systematic artifacts. Okay? And what you do is you have a panel of normals. So not your patient. Get different participants, different patients, sequence with the same sequencer as much as consistently as possible, and look to see if they have the same artifacts. And I'll show you this tomorrow. But you compare other data with yours to say, look, if I see the same signal in this other data, it's clearly not unique to my sample. It's an artifact. Okay? So we'll give you lots of examples of that when we talk about it. Here's one more example you can kind of see, but all the way on the left on the bottom, you see that blue column. That could look like a heterozygous SNP. That's what it looks like in IGV. And you don't see anything in the matched normal. Whereas for the other stuff in the tumor, you do see things in the normal. So probably those are not somatic, those are germline. Questions about that? And here's an example of an artifact. Okay, you can see in these, through the genome, things that can trick a variant color. They're just not great sequencing. I'll say IGV. We mentioned that when when a base is different than the reference, it has a color, but the intensity of that color is proportional to the confidence, the quality score. So if it's a Q40. 99.99% confident, it's bright blue. But if it's a Q10, only 90% confident, well, it's kind of faded. So you see there's a lot of faded things around here. So we use that as signal to say, look, this is not real. I see the same thing in other samples also, so we'll remove it. And we will do a lot of this on Wednesday. Somatic CNV. So this is copy number variants, larger mutations. This example here actually shows P53. It's a very important gene in the cancer world. TP53 is in charge of apoptosis, cell death. When a cell is working well, nothing happens. But when a cell is misbehaving, TP53 says, all right, time to turn off and kill itself. And so also in this example, actually, you see that TP53 has 5 copies instead of the 2 you'd expect. But there's also mutations in there. So the bad thing, so it becomes an oncogene, a bad gene, when TP53 is turned off. You don't want it turned off because then the cells aren't telling it to kill itself. And so this is one of the most studied genes in the somatic space. But to know how many copies you have is important. So MDM3, I believe, or MDM2 is the gene that regulates TP53. So if you have 7 copies of MDM2 and still have 2 copies of TP53, you've got a problem because now TP53 is being overregulated. That makes sense? Yeah. So knowing the copy number of these genes that are all affecting and interacting with one another is crucial for the understanding of cancer. So we will teach you on Wednesday all about how to do this. It's very complicated because, so remember, so a lot of the cancer sequencing is done at this exome, because again, you really mostly care about the genes and how those are affected. So the exomes we talked about being inconsistent. Any one exon is very different from exon to exon. So when we talk about both cancer and in germline CNV calling, very difficult to do that because there's a lot of inconsistency across the depths, the amount of sequencing you see in any one exon, even within a gene. Okay, and some of the events are big and some are small, and some of them are germline. It's very hard to tell what is signal and what is just this exon doesn't capture well, or the particular. So the Broad adds some extra for some exons that they like. They add some extra capture inside. So arbitrarily, the Broad is capturing differently than you do here in Taiwan. Okay, so all of that is very difficult, and we have to we deal with that computationally. Okay, and we'll teach you all about this on Wednesday, all the processes and math and stats involved. Okay, and then on Thursday, you'll have Emma teach you our latest offering, which is now the structural variation. Structural variation is super complicated. So I think GCNV, the germline CNVs will happen on Wednesday afternoon. It is, again, very similar to the somatic use case, trying to figure out where where these copy numbers are. Most of the mutations in cancer are very big. Whole arms of chromosomes are missing. We have a cool picture coming up on, I'll show you on Wednesday, of a cell that is a somatic breast cancer cell or a colorectal cancer cell. And cancer does terrible things to your genome. Okay, but in the germline case, if you have made it to adulthood, most likely you have CNVs, but they're not that crazy. They're usually smaller and don't affect too many cells. Otherwise, you wouldn't be a viable embryo. Okay? So because these are smaller, again, you have an exome. So you're only seeing coverage every so often, and you have these small events. They're very hard to detect. The signal is very small. Not a lot of noise necessarily, but not a lot of signal either. And so it's a whole different problem. And we'll teach you all about that on Wednesday. This is kind of a schematic. I think either Emma has this picture or a much better one in her presentation. Structural variation is complicated. Some really crazy things happen. It's not when there's an inversion, so there's a whole swath of your DNA that's turned around. It's not usually just an inversion. There are other things. There's an inversion and a duplication next to it. Really crazy things happen in the human genome. And so detecting these structural mutations is very hard. So that is something where you'll be our guinea pigs to test how our module is. And you can tell Emma how she did in preparing her teaching for you on Thursday. Okay, so I think we are done with the overview here. I just want to say for almost everything we have should be present and available for you in both in GitHub. Okay, you should be able to take a look at this. Again, you have access to all these slides. So if I go too quickly, just go back to the slides and look at them on your own time. If you want, you don't have to write down web URLs right now. You can always go back to these and look them up. But all of these are available on GitHub. I don't know about the SVs yet. You can ask Anna when she's back in the room. That is for sure available in Terratin. And again, as we pointed out, a lot of these things will be not only available in Terra, but available in the workspaces that that are featured. So you can easily go to them. Anton taught you all about how to access them. And that is that for an overview of variant discovery. I'm gonna pause for a minute for just to take questions if anyone wants to ask questions, and then we'll jump into specifically germline calling. So for SNPs and indels with the HaplotypeCaller. So just any questions in either room? Don't all jump up at once. I'm going to just wait in case anyone from the other room comes. Go. All right, Aaron left. So, all right, I'm going to go then. Okay. So the HaplotypeCaller is the name of the tool we use to detect germline, SNPs, and indels. When we talk about somatic, the tool is called Mutect2. It's actually built on top of the HaplotypeCaller. So all the stuff we're going to learn about today is relevant to that too. And for those of you who have heard about Dragon GATK, so there's actually you will have heard all about this tomorrow or Wednesday. But let me just say that everything that's part of Dragon GATK is inside the HaplotypeCaller already. So all of this is still relevant, just to be crystal clear. I'm going to give you a high-level overview of it. If you'll hear about this again, but just so you understand this, the GATK is open source. So multiple different companies took our open source code, as they are allowed to, and made it faster, different, and some of them have shared back differences we've collaborated with. So that's in particular the Dragon. So you'll learn all about what changes went into the HaplotypeCaller tomorrow or Wednesday. Now let's just focus on the high-level overview of this. Okay, so we're going to do that top middle section now. So what you heard earlier from Emma was how you go from the raw data that comes off the machine, you align it, you remove duplicates, you do base quality score recalibration to fix the quality scores. So now you have a BAM file that's ready to be analyzed. So what do you do? Okay, there are 4 distinct. I will go through the details of each of these steps, but there are 4 steps in a high level for the HaplotypeCaller. We'll go through each one of those. Step 1, we call identify active regions. Now this is completely to make it for speed and optimization. We could do so that all the stuff in the next 3 steps are complex computational math. Okay, we're going to actually show you some of the math. I won't go into too many of the details. I won't scare you with the math, but there's a lot of math. And so you're doing that at 3 billion bases. That takes a long time. But remember from your IGB screenshots, most of the time there weren't any SNPs there. It was just gray bars. Well, why should I do all this math on the gray bars when I know nothing is going to come? So what we do first is we do a quick pass every so often and say, do I see what we call entropy, energy, any differences? And if I see enough differences in a region, I'll say, oh, okay, now I'll spend my time to do the compute here. But if I don't see any differences, let's just not bother and we'll keep going. And that's the point of active regions. It's just to make things go faster. Then the next piece here is I say every so often I found now an active region. So I found in IGB, we don't do this in IGB. This all happens in inside the GATK. But imagine in IGB you see a region with lots of differences. So I stop and I take the reads from that region. I ignore the rest of the genome right now in this one active region. When I take all those reads that were aligned before with BWA or whatever, and I'm going to realign them. I'm going to do a new thing with them. I'm going to not trust BWA anymore. BWA got us the reads in the close to the right location, but I don't like exactly I don't trust it necessarily. And I'll show you some examples of what. So I'm going to go ahead and assemble the reads. Now assembly is different than alignment. Alignment says, I see a reference. I'm going to try to find where in the reference it matches. Assembly says, I don't have a reference. I'm just going to try to find similar k-mers inside these reads. Okay? So you can do that with split quantum. You can do that other things with graphs. What we usually do is we create a graph. The HaplotypeCaller uses a De Bruijn graph. The details are not important. But let me kind of show you what it looks like. So imagine, so actually the reference is technically a path in the graph. So in our toy example, on the top, I have the reference sequence ACPTAGA. So that's now we have a graph, visual graph. And you can imagine a path to the graph. It's all single direction, unit direction. Okay, so it's no cycles in this graph. And as we see a new read, let's say we see a read that says ACTGAGA. So I put it into the graph, and I notice, now you should notice in the second line, there's a split after the T, the first T, and you can take two different paths in the graph because the reference takes one path, and the read we saw takes a different path. Yeah? This is how you do alignment or assembly. And at some point, I'll throw reads in, and imagine this is the case of a heterozygous SNP. So half the reads will have the reference allele, the wild type allele, and half the reads will have a mutation. So you'd expect, let's say there were 7 reads you saw, maybe half of them, 4 had the T and 3 had the G. So you count those in your path, and you've done, congratulations, you've done assembly. Pretty simple? Any questions about that? All right, so this is an example of why we don't trust BW. BWA, again, was good, but this is why we don't. If you look on the top, this is actually, let's look on the bottom and explain why. Look at the reference sequence. If you look carefully, you see AG, 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 TG, 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 over and over again. So this is exactly the case that the sequencers do terribly on. Once it sees too many A's and G's or P's and G's, it starts to hallucinate and imagine the wrong bases. It just, I don't know why, the chemistry starts to bleed and there's issues. Right? And so in this case, also biologically, this is also the case where the human genome messes up. And this is how, PCR, everything happens. You have slippage in the biological mechanisms. And so there were actually truly 2 deletions here. But if you look up top, there's deletions of 8 bases, 10 bases, 12 bases, 7 bases. There's lots of those discolorations. That means they're SNPs, but they're all in a row. What's going on there? That's because BWA only sees one read at a time. So it does its best with that read. And it knows it belongs in this general region, but doesn't know exactly how to align it. But when we see all the reads together, we can be a lot more informed and smarter about it. And as we start putting them into this graph, we say, look, the truth is there's 3 paths in the graph. There's the reference. There's a 1 deletion with 12 bases and a 1 deletion with 10 bases. And every read is consistently categorized with just that, those 3 paths in the graph. It's pretty amazing. Another thing that happens occasionally is you can have 2 different mutations that mean the same thing. Or maybe a better way of saying that is there are 2 different ways to describe the same mutation. So in this example, look at the top, that top right, the top path. You have a T in the reference that gets turned into an A and a C. So it's like 1 base is different and 1 base is inserted. But how do you describe that? So this is a true alignment for BWA. These same reads, one of them has an insertion of an A, and then a C, the T gets turned to a C. See that on top? And reads below it, the T gets turned to an A, and there's an insertion of a C. But that's the same thing. They actually translate to the exact same thing. But BWA doesn't know that. It just does its best. But when we see all together, we see, wait, these are the same thing, and there are only 2 paths in this graph. The reference and the change from the T to the AC. And so we can better understand and quantify all the alignments and all the mutations. This would look like a heterozygous mutation, but actually it's homozygous. Every read has it. And one other important thing with using the graph, this is very important for understanding how mutations can affect their samples. is you can phase. Phasing means you could take multiple mutations and tell me if they're on the same haplotype. Haplotype is just a swath of a region of DNA. Right? So you're here looking at the left example. You notice that there's two heterozygous SNPs that are both on the same haplotype. If you have one of them, you have the other one too. So if you assume you can get one from your father, you got the other one from your father too. Okay, but that means that the other copy from your mother is completely wild type, completely normal. Whereas on the right, if you have the C SNP, the blue one, means you don't have the red one, the T. Now, why is that important? Let's say both the C and the T on the right were major differences that affected the amino acid that was produced by that gene. Well, now you have two bad copies. Both the copies from your father and your mother are affected here, which means you have two bad copies of the gene. Okay. Whereas on the left, you might only have one bad copy, so you could only be a carrier, right? If it's a recessive. So phasing is very important. So this is called cis on the left and trans on the right. By doing this graph, we actually can see all that. If you only look at one thing at a time, you can't. All right, so that was step 2, creating that graph. Now we're going to use it to start doing some math. So bear with me a little bit here. So let's look at the case I have here. So let's going to look at 4 possible haplotypes. I want you to make sure you understand it. So I have a new toy example here. So there's 2 potential SNPs. An A turning to a C on the left, and a T turning to a G. Okay? And so the 4 possible haplotypes are listed to the right, middle right. Notice the red. I can be on the number 1, I have an A and a T. That's the reference. Okay? These are the possible ways I can go through the graph from the start to the finish. There's 4 ways I can go. Top and top, top and down. That's 2. I can go down and up, and then down and down. There's 4 ways. Now, that doesn't mean every read, that every possible haplotype is seen in the reads. But that's the 4 possibilities. So now we look at the reads. My example here, I have 3 reads. Notice read 1 has a C goes down and a T goes up to the second one. The second read has a C and a G. And the third one has an A and a T, but there's another T also. Okay? It's an error. It happens. I said 1 in 1,000. Now it's 1 in 30. So we then have to score each of the reads for each of the haplotypes. So let's just take the first read as an example. So read 1, again, has a C mutation and a T mutation. We're going to go look at row 1 of that bottom table. Those numbers, we're looking at the exact row 1. So they should not all add up to 1. These are not probabilities. What's going to be in this row are the likelihoods that that read is true, given that the haplotype is true. So if the haplotype 1 was the real haplotype that read 1 belonged to, what is the likelihood that's the case? Now, it's not a probability, so these things don't have to add up to 1 yet. Okay, but it's not a non-zero possibility. Okay, the likelihood is not 0. There could be an error. It's possible. So again, there's a C and a T in the read, and an A and a T in the first haplotype. Make sense? So how is it possible if there's a C and a T in the read and an A and a T in the haplotype that could? Well, what if the C was a mistake? What if the C was one of our errors? That's how. So it is still possible that the first read was present from the first haplotype. So there's the likelihood we're calling, I made this number up, is 0.6. Not 60%, it's a likelihood. How about second haplotype? Second haplotype is an A and a G. And ours is a C and a T. That's really unlikely. Not non-zero. It's still 0, not 0, but we're calling it 0.01. Now the most likely scenario, though, 0.99, not percent, just likelihood, is that the first read, which has the C and the T, came from the third haplotype, that's the C and the T. And all these, I'll talk about how these are scored, but the most important thing is we asked before about base qualities. The scores here are all affected by the base qualities. The base qualities are low, my scores stay low because we don't have any confidence. But if the base qualities are high, we get some very high scores. Okay, questions about this? This is some of the hardest stuff. I can always review this again later. Yeah, ask the question.
I know why the that's why the magnitude is very much more higher than the two.
Okay, so for haplotype 2,
Yeah.
why is read
Three
3
have higher magnitude than the race two.
then read 2? Okay, so remember, everything depends on the base qualities, and this is a toy example. But read 2, so haplotype 2 has an A and a G. Read 2 has a C and a G.
Yep.
Okay, so we have that number there. Read 3 has an A and a T and a T. So there's 2 differences on that one. But who knows if that difference could be low quality. Could be that this base was a Q10, whereas it matches it on the A, the first A. That A could be a Q40. And that's the difference. That's where the base qualities come in. Okay, so it's a toy example. It's a good question, a toy example. But these things really happen. Other questions? It's in the afternoon now. Math is hard. I know math is hard. We got this. Okay. So we always take, although we're looking at all the mutations together, we score them one at a time. So let's just look at the leftmost mutation. Okay, so the AC. So we're pulling out the AC data from that table. And so for each of the reads, I'm taking the highest score. So just believe me, because I've done this for us. I've taken the highest scoring A and C alleles scores for each one of those things. I can go through this if you want, but just trust me. And I pulled out the highest scores. For each of the reads, the highest C score for 1 is 0.99, and for A is 0.6. So that's now what I'm going to look at. Okay, and I'm going to throw this into our math. And I'm not going to make you understand every bit of detail of the equation, but this is simply Bayesian mathematics. It's Bayes' law. Okay, so I'll actually jump down to the bottom to start with. Bayes' law is the posterior probability. Now that adds up to 1. Probability is just the prior probability times the likelihood. Okay, so what we do at the Broad is we assume no prior likelihood. So and it ends up being that our likelihoods do become probabilities. I lied to you, but for now, it's not true. So I know this is a lot of math, but your prior probability is what do I know in advance about this genotype? Well, let's say I knew the parents' genotypes. I would have a strong prior probability, right? It doesn't mean I know it for sure because there are de novo mutations. If I know something about, or if I know it's an Asian sample, well, I can look at the frequency in the population and say, look, there are many people with the same mutation in the population, so I have a higher probability that this is a real mutation here. If I don't know anything, right, they're any possibility is equally probable. We'll talk a little bit about this tomorrow with genotype refinement. Okay, but everything here is assuming diploid genomes. That's what the divided by 2s are there. And we're looking at really the possibility of any one of these reads that they exist based on the possible genotypes, A's and C's. And again, we're allowing 2 possibilities here because there are diploid genomes. And so all that math goes in and we get answers. So in my example here, the top row on the left is your likelihood. So I have these numbers. And I said, remember, your likelihoods are not going to add up to 1. Okay, this is all the math that goes into it. So in this example here, imagine the possibility that someone is A in an A is, this is like likelihood 0.032. And same thing for CC is also even lower. But for AC, it's 0.385. Well, so that's going to be the more likely possibility. When we represent this in a file, using small numbers like this is really inefficient to store. It does not compress well. So what we often do is we red scale it. We turn it into integers. Integers, they compress really, really well. And they're much more easy to understand in that sense. So we always Fred scale our likelihoods. And now Fred scaling, we talked about this, the negative 10 log 10. So so the next row flips them down to raw Fred scaled likelihoods. Okay. The next thing that we want to do, so again, that raw PL is again PL is a Fred scaled likelihood. That's how we represent the math inside the file. But 14.95 and 4.11, that also doesn't, we lost our integerness. So what we do is we normalize it so that the most likely one, because we've done negative 10 log 10 of the error, the lower number is better. The lower it is, the better it is. And this is all log math. Okay. So we just make sure the lowest number becomes 0 and everything else is normalized to that. So in the file, and then we make everything integers. So everything now in your file is an integer. So what you see truly in your file is 10, 0, and 20. That means the more likely possibility for the sample is AC, because it's the 0. The 0 is good. The lower, the better. Okay? So the difference between that one and the next best one is 10 on the left, the AA. So that we say that our genotype quality, our genotype confidence is 10, Q10. The difference between the best, the most likely, and the second most likely is 10. And that's, if it was 100, we'd say we have a Q100 confidence because they're so far apart mathematically. But this is far apart, by one order of magnitude. 10 is a Q10. Okay, so we're only 90% more confident in one over the other. Oh, that's still a lot. When we see 30x coverage, usually you see genotype quality is like 99, which is the max. So you're 99.999999% confident that this is the right genotype. We still make mistakes, but yeah. Okay, this is pretty complicated. Questions? Yeah.
How much of genotype inequality is acceptable?
How much of genotype quality is
Acceptable.
settable?
Acceptable. Acceptable.
Acceptable.
Yeah.
Oh, great question. So the question is what is the cutoff really?
Yeah.
What's a good genotype quality? And it all depends on what you're doing. If you're looking at de novo mutations, so changes that are different from a child parent, you only expect 20 of them, the whole genome. So if it's below Q60, probably don't trust it. Okay. If you're looking at, did I get it? Is it a homozygous reference or not? Q30, Q40 is fine. Okay. So it really depends. What most people do is they set a cutoff around Q40. That's still pretty good. But again, you'll also see for most of the sites, most of the genotypes will be Q99. Good question. Other questions? Okay. Okay, so that was the HaplotypeCaller. I mentioned also that there is a mode to skip the last step. So the last step assigns a genotype, but we might not want to do that. We want to do joint calling, so we might want to kind of assign a genotype, but not finish the process because we want to do the whole population level, all the samples together. That Aaron will talk about at the end of the day today. So we kind of we create, instead of a VCF file, something called the GVCF file. Again, Aaron will talk about that. But it's most of the way over to variant calling, but not quite to the finish line, so that we can then put all the samples together in memory and determine what everything is together. And to do that, we need to set up use a different argument. I'll show you in a second, and then run one of our joint calling modules to group all the data together and analyze them together. So on the top, you'll see the basic way of running HaplotypeCaller, which we're about to do in like 2 minutes. Okay, so everyone get excited. run the GATK. The tool is called the HaplotypeCaller. And because this is the first time we're doing this, I'll kind of take just a second to show you that the GATK has different arguments. So that -R means the reference file. So mostly with the GATK, you'll want to provide a reference because all the reads are aligned relative to the reference. So we need to actually use it in all of our analyses. -I is the input BIM, and -O is the output VCF. Pretty simple, okay? Reference, input BIM, output VCF. When we want to do the population-based calling, the joint calling, we add one argument. So instead of being done at -O, we don't create a VCF, we call a GVCF, okay? And then we add the GVCF mode. And Aaron will explain to you, you'll see in the hands-on, what is different between the GVCF and the VCF. And that is that. So we'll do joint calling after the hands-on. Right now, we have the hands-on exercise that Aaron will run. While Aaron comes up, and I'll let him introduce himself also, I'll take questions both either from this room or the next room, because I'm done up here. Thing. We'll just give it 1 minute to see if anyone comes to the next door. Sure. So this is. You actually do one. Oh, question. Great question, man. One to go. Sure. I'm gonna keep the microphone. Okay.
Hi, Eric. Hi, team. I have one question about the HaplotypeCaller. So you put a lot of resources to assemble the haplotypes and do the output of our VCF file, have the information of the haplotypes, because we need the haplotype in the regional scale, not the global scale. It is enough, but do have the information. Thank you very much.
That was a great question. Great question. I forgot to mention this as one of my slides. It was there. There is a mode to output the haplotypes. It's called BAM out. The BAM. So we talked about the flags. -R is for the reference. -I. There's also -BAM out, and that will let you output the alignments. You can see them, and you can see the original and the new haplotypes, and the reads aligned to the other haplotypes if you want. It's not on by default because it's a lot. It's a lot of practice. You have to do a lot. The input and output is the thing killing you here. You're outputting reading in all the reads and reading out all the reads again. And so most people don't want to spend the compute for that, but you can see it for sure. Great question. Did I answer your question?
Yes.
Yeah.
Thank you very much.
Yeah, of course. Anyone else? Just going to let Aaron get set up here. So Aaron will run for the next approximately 1 hour. Then we'll take a quick break. We'll do joint calling, and then you get your stickers.